Higher organisms, such as animals, must acquire alternative metabolic pathways that create more energy than glycolysis to have the ability to swim, run, and jump. Thus, became possible once plants covered the earth, thereby filling the atmosphere with oxygen, which is a product of photosynthesis. Thus, the possibility for large-scale oxygen-dependent aerobic respiration was born. Aerobic respiration is the process of producing cellular energy involving oxygen. The location in the cell where the process happens or occurs is in the mitochondria. Mitochondria is entirely separate organisms that were incorporated into eukaryotes, specifically for the respiratory abilities they possess, according to endosymbiotic theory. The process starts with the pyruvate molecules that is produced in the cytoplasm during the glycolysis. This pyruvate will enter the mitochondrial matrix to find the coenzyme A. In the presence of NAD+, pyruvate will undergo decarboxylation, oxidation by the help of NAD+, and the attachment to coenzyme A generating acetyl-CoA. Acetyl-CoA will then enter the citric acid cycle, also known as the Krebs cycles or tricarboxylic acid cycle.
Alternate titles. The citric acid cycle is also called the tricarboxylic acid cycle and the Krebs cycle. Citrate synthase. The first stage of the citric acid cycle is catalyzed by the enzyme called citrate synthase and requires acetyl-CoA and oxaloacetate as substrates. Citrate. During stage 1, the 2-carbon acetyl group from acetyl-CoA is transferred to the 4-carbon oxaloacetate to form a 6-carbon citrate and CoA. Aconitase. Stage 2 of the cycle is catalyzed by the aconitase enzymes and begins with citrate as a substrate. Stage 2 involves true structural changes to the substrate. First, a water molecule is removed from the citrate compound to form cis-aconitase. Finally, the cis-aconitase is rehydrated to form isocitrate. Isocitrate dehydrogenase. Stage 3 of the citric acid cycle is catalyzed by an enzyme called isocitrate dehydrogenase and begins with isocitrate and NAD+ as substrates. Ketoglutarate. The oxidation and the carboxylation reaction of stage 3 result in the production of 1 carbon dioxide, 1 Ha+ ion, and 1 NADH, and a 5-carbon A ketoglutarate. Stage 4 of the cycle is catalyzed by a ketoglutarate dehydrogenase enzyme and begins with CoA, NADE+, and a ketoglutarate as substrates. Stage 5. In the 5th stage of the citric acid cycle, an enzyme called succinyl-CoA synthetase catalyzes a reaction between GDP-PI and succinyl-CoA. This reaction results in the production of CoA, succinate, and 1 molecule of the GTP, and GTP will always be converted into ATP. The 6th stage of the cycle is catalyzed by succinate dehydrogenase enzymes and requires succinate and FADH as substrates. This enzyme is embedded within the membrane of the mitochondrion. Fumarate. The resulting products of this oxidation reaction are fumarate and 1 molecule of FADH2. The FADH2 can be used later to create more ATP for the cell. In the seventh stage of the cycle, a fumarate hydratase enzyme catalyzes a reaction between water and fumarate. Malate dehydrogenase. This reaction results in the formation of malate. In the eighth and the final stage of the citric acid cycle, a malate dehydrogenase enzyme oxidizes NAD+ and malate. This reaction regenerates the original oxaloacetate molecule from the first stage of the cycle along with one NADH and one HA ion. From glycolysis, for every one molecule of glucose that enters glycolysis, two acetyl-CoA molecules will be produced. This acetyl-CoA molecule then enter the citric acid cycle. Alternate substrates, in addition to sugar like glucose, proteins, and fats can also enter the citric acid cycle at various stages. Proteins can be broken down into intermediates that can enter the cycle, and fatty acid can be broken down into acetyl-CoA, which begins the citric acid cycle. Now we will discuss the citric acid cycle 8-step pathway. In the first step, enzyme citrate synthase removes the acetyl group and taxidon to oxaloacetate to form citrate. Next, with the help from aconitase, a water molecule is removed and another one is added to generate structural isomer of citrate called isocitrate. Then, catalyzed by isocitrate dehydrogenase, isocitrate is oxidized NAD+ and decarboxylate to form alpha ketoglutarate. Next, another CO2 is lost and further oxidation by NAD+ takes place with the help of ketoglutarate dehydrogenase. The resulting molecule will join with coenzymes once again to form succinyl-CoA. CoA and then displaced by phosphate group to form succinate, which is catalyzed by a succinyl-CoA synthetase. This will make one molecule of guanosine triphosphate or GTP in the process, which can be used to make one ATP with the help of succinate dehydrogenase. Succinate oxidized by a different molecule FAD, which result to fumarate and FADH2. Next, fumarase will catalyze hydration, which result in malate. And lastly, one move oxidation NAD+ takes place with the help of malate dehydrogenase to give oxaloacetate, which will restart the cycle reacting to the new acetyl-CoA. Overall, for every acetyl-CoA that enters this cycle will produce 3 NADH, 1 FADH2, and 1 ATP, since 1 glucose will produce 2 pyruvate glycolysis and therefore 2 acetyl-CoA. We can double these numbers to get the amount per glucose molecule.
The cellular respiration pathway begins with the citric acid cycle and then goes through glycolysis. Both processes generate ATP, but the ATP from the FADH and NADH pathways is far more potent than the other ATP sources. This pathway utilizes a series of mitochondria membrane proteins known as the electron transport chains. They are situated in the inner membrane of mitochondria. Ubiquinol is a small hydrophobic molecule that situated with the membranes of the cell, and it is known to be coenzyme. These components of the electron transform change is also responsible from the generation of ATP sitase protein complex.
The protons move spontaneously along the gradient to re-enter the mitochondrial matrix. The only available way is ATP synthesis. These protons drive the ATP synthesis phosphorylate ADP to produce a water jet that rotates by the water wheel, which can then be a different process for each DHNF ADH2. It is produced by a single glucose molecule, most of the cellular energy. So, in a nutshell, your body uses multiple metabolic pathways for glycolysis. The oldest one produces 2 ATPs for glucose, but it also causes pyruvate, which can be here entering the citric acid cycle. These two pyruvates will produce another 2 ATP, but they will also produce 6-NADH and 2-FADH2 molecules, which can enter the electron transfer chains, thus providing us with large ATP and only one glucose molecule. In addition to glucose, other food sources such as protein, fat, and other carbohydrates were originally based on the unique decomposition of waste C, but this always leads to compound that eventually enter one of the metabolic pathways we are discussing. So all the food we eat ends up. They are always broken down in this way, which is why mitochondria are considered to be the motors of cells, as most of the energy required for cell activities come from them.
Chemiosmosis. ATP synthase is the component that synthesizes ATP. When the intermembrane space is greater concentration than inside the mitochondrial matrix, the protons will spontaneously move with a gradient to re-enter mitochondrial matrix, and the only route available is through ATP synthase. This process is called chemiosmosis. If the gradient is able to do work through chemiosmosis, we also call this as proton motive force, in which proton power the ATP synthase in phosphorylating ADP to generate ATP. ATP synthase. ATP synthase has a fascinating structure that looks like a rotor, where individual protons can bind and cause it to spin in such a way that catalyzes phosphorylation ADP. The process in ATP synthase is like a stream of water turning a water wheel, which can power some process. With NADH and FADH2 that is being generated from a single molecule of glucose, around 26 to 28 of ATP is being produced from this activity. Foods beside glucose eventually feed into cellular respiration. Other sources of food like proteins, fats, and other carbohydrates beside glucose are initially broken down in unique ways. All the food we eat eventually gets broken down. We regarded mitochondria as the engine of the cell, since most of energy needed for cellular activity comes from them.